Assalamu alaikum friends. Hope so that you all will be fine. Friends, in this video I am going to discuss about an important topic of carbohydrate metabolism, that is gluconeogenesis. We will cover this topic under the heading of definition, important aspects, site, steps and regulation. Okay. So first of all, let's start our lecture with the definition of gluconeogenesis. The definition is the synthesis of glucose from non-carbohydrate precursor. Okay, the synthesis of glucose from non-carbohydrate precursor, it is called as gluconeogenesis. The non-carbohydrate precursor includes lactate, glycerol, propionyl CoA, and glucogenic amino acid. Okay. Now coming toward the important aspects of gluconeogenesis. It is not complete reversal of glycolysis. Only three steps they are reversed by the specialized enzyme of gluconeogenesis. So important aspects. First one, it is not a complete reversal of glycolysis. Okay. The second one is the 6 ATPs are utilized in the synthesis of per molecule of glucose. The third one is it is an important source of glucose in starvation. In starvation, it is an important source of glucose. The fourth one is it is an important source of energy for brain and matured RBC. It is a source of energy for brain and matured RBC. Okay, these are the important aspects of gluconeogenesis. Now coming toward the site for the process of gluconeogenesis. Gluconeogenesis process occurs in liver and renal cortex. Okay, in liver and renal cortex. There are two important organelles required. First one is cytosol and another one is mitochondria. Okay. Now coming toward the steps of gluconeogenesis, as discussed that it is not complete reversal of glycolysis. So let's see these three steps, first, second and third. These three steps, they are reversed by the specialized enzyme of gluconeogenesis. There are four important non-carbohydrate precursors that are, first one is lactate, second one is glycerol, third one is propionyl CoA, fourth one is glucogenic amino acid. Before the discussion of the gluconeogenesis from these four non-carbohydrate precursors, let's see the common pathway. In this pathway, we discussed that how pyruvate converts into the phosphoenolpyruvate. How this irreversible, sorry, this is a irreversible reaction. How this converts into the phosphoenolpyruvate. Okay. Now let's discuss the common pathway. Okay. Now the conversion of pyruvate into phosphoenolpyruvate. This pyruvate, with the help of pyruvate carboxylase enzyme, it requires ATP, biotin, and carbon dioxide. With the help of pyruvate carboxylase enzyme, this pyruvate it converts into oxaloacetic acid. This oxaloacetic acid first the mitochondrial membrane are impermeable for this oxaloacetic acid. So this converts into malate. Now malate easily crosses the mitochondrial membrane and in cytosol it converts into the oxaloacetic acid. In cytosol it converts into the oxaloacetic acid. The enzyme required, that is malate dehydrogenase enzyme. Okay. Now then oxaloacetic acid in cytosol it converts into phosphoenolpyruvate with the help of phosphoenolpyruvate carboxykinase enzyme. It requires GTP and there is removal of carbon dioxide. Friends, it is a common pathway how pyruvate converts into the phosphoenolpyruvate, while all other steps they are all reversed by glycolysis enzyme. Pyruvate it converts into oxaloacetic acid with the help of pyruvate carboxylase enzyme. It requires ATP, biotin, and carbon dioxide. The mitochondrial membrane they are impermeable for oxaloacetic acid, so it converts into malate with the help of malate dehydrogenase enzyme. Malate easily crosses the mitochondrial membrane and cytosol it converts into oxaloacetic acid, and oxaloacetic acid converts into phosphoenolpyruvate with the help of phosphoenolpyruvate carboxykinase enzyme, and then phosphoenolpyruvate converts into the again same enzymes of glycolysis. It converts into the now here this fructose 1,6 bisphosphate converts into the fructose 6 phosphate with the help of fructose 1,6 bisphosphatase enzyme. Okay, this is the specialized enzyme of gluconeogenesis, and this glucose 6 phosphate it converts into the glucose. This glucose 6 phosphate converts into glucose with the help of glucose 6 phosphatase. Okay, this glucose 6 phosphatase, fructose 1,6 bisphosphatase, phosphoenolpyruvate carboxykinase enzyme, and this pyruvate carboxylase enzyme, they are the important enzyme of gluconeogenesis. I am repeating again, the pyruvate converts into the oxaloacetic acid with the help of this pyruvate carboxylase enzyme. It requires ATP, biotin, and carbon dioxide. This oxaloacetic acid first converts into malate, and malate easily crosses the mitochondrial membrane, and in cytosol it converts into oxaloacetic acid, and in cytosol oxaloacetic acid converts into phosphoenolpyruvate with the help of phosphoenolpyruvate carboxykinase enzyme, and they are in this now here this fructose 1,6 bisphosphate converts to fructose 6 phosphate with the help of fructose 1,6 bisphosphatase enzyme, and glucose 6 phosphate convert into glucose with the help of glucose 6 phosphatase enzyme. This glucose 6 phosphatase enzyme it is present in liver and kidney. It is absent in muscles and adipose tissue. Okay. Now, how lactate converts into the glucose? Lactate, first lactate it converts into pyruvate with the help of lactate dehydrogenase enzyme, and here NAD plus, it converts into NADH and H plus, and it is a reversible reaction. The conversion of lactate to pyruvate, it is a reversible reaction. And then pyruvate, now let's see here, pyruvate, then it converts into oxaloacetic acid converts into phosphoenolpyruvate, and then again here fructose 1,6 phosphate converts into the fructose 6 phosphate with the help of fructose 1,6 bisphosphatase enzyme, and glucose 6 phosphate converts into glucose with the help of glucose 6 phosphatase enzyme. In this way, lactate forms glucose. In this way, lactate forms glucose. Okay, first of all, lactate converts into the pyruvate and is a reversible reaction, and in this way, pyruvate converts into oxaloacetic acid, and then again, same reaction. Okay, now second one is from the propionyl CoA. Second one is propionyl CoA. This propionyl CoA, it converts into the methyl malonyl CoA with the help of propionyl CoA carboxylase enzyme. Propionyl CoA carboxylase enzyme. This requires ATP, biotin, and carbon dioxide. And methyl malonyl CoA, in next reaction, it converts into succinyl CoA, which is the intermediate of TCA cycle. And succinyl CoA, in next reaction, it converts into the oxaloacetic acid. Okay. Here, methyl malonyl CoA converts into succinyl CoA with the help of mutase enzyme, which requires vitamin B12. And oxaloacetic acid, again here, oxaloacetic acid converts into the malate because mitochondrial membrane are impermeable for oxaloacetic acid, and malate, it crosses the mitochondrial membrane, and cytosol, it converts into the oxaloacetic acid, and oxaloacetic acid in cytosol converts into the phosphoenolpyruvate with the help of phosphoenolpyruvate carboxykinase enzyme, and again the same steps are repeated. In this way, propionyl CoA involves in the synthesis of glucose. First, it converts into the methyl malonyl CoA with the help of propionyl carboxylase enzyme, and then it converts into succinyl CoA with the help of mutase enzyme, and succinyl CoA, which is the intermediate of TCA cycle, it converts into oxaloacetic acid, and then oxaloacetic acid converts into the phosphoenolpyruvate, and then into the glucose. Okay. Now coming toward the synthesis of the glucose from glycerol. So this glycerol, this glycerol with the help of glycerol, with the help of glycerol kinase enzyme, it is converted into glycerol 3 phosphate, and glycerol 3 phosphate in next reaction converts into the dihydroxyacetone phosphate with the help of glycerol 3 phosphate dehydrogenase enzyme, and in this way the dihydroxyacetone phosphate converts into glucose. So glycerol converts into the glycerol 3 phosphate with the help of glycerol kinase enzyme, and glycerol 3 phosphate is converted into dihydroxyacetone phosphate with the help of glycerol 3 phosphate dehydrogenase enzyme, and the same steps, and they convert into glucose. In this way, glycerol forms glucose. Now coming toward the synthesis of glucose from glucogenic amino acid. The example of glucogenic amino acids are, first of all, glycine, serine, and cysteine. This glycine converted into the serine Serine, it is converted into the pyruvate with the help of serine dehydratase enzyme. Serine dehydratase enzyme, it is converted into the pyruvate, and again this pyruvate converted into oxaloacetic acid is converted to phosphoenolpyruvate, and then the same steps. Okay, again the same steps. I'm repeating again. This glycine converts it into the serine. Serine converts into pyruvate with the help of serine dehydratase enzyme, and pyruvate it forms the glucose by this way. Pyruvate converts into oxaloacetic acid and phosphoenolpyruvate and then glucose. Now coming toward the synthesis of the glucose from serine. Serine it forms pyruvate with the help of serine dehydratase enzyme, and again the same steps are repeated. Pyruvate converts into oxaloacetic acid and phosphoenolpyruvate and then glucose. Now last one is cysteine. Cysteine it converts into the pyruvate with the help of cysteine desulfhydrase enzyme. Cysteine desulfhydrase enzyme, it converts into the pyruvate, and then pyruvate converts into oxaloacetic acid and then phosphoenolpyruvate and then into the glucose. In this way, the glucose gets synthesized from non-carbohydrate precursor. First one lactate, another one propionyl CoA, third one is the glucogenic amino acid, and then glycerol. This is the common pathway. This is a common pathway because first, second, and these three steps they are reversed by specialized enzyme of gluconeogenesis. And how pyruvate converts into phosphoenolpyruvate? Let's see here. Pyruvate converts into oxaloacetic acid and phosphoenolpyruvate, and then into the glucose. These are important enzymes: glucose 6-phosphatase enzyme, fructose 1,6-bisphosphatase enzyme, pyruvate carboxylase enzyme, and phosphoenolpyruvate carboxykinase enzyme. These are important enzymes of gluconeogenesis. Now coming toward the regulation of gluconeogenesis. Okay, regulation. Regulation. The regulation takes place by three ways. First one is through hormones. This insulin it inhibits the pyruvate carboxylase enzyme. In this way it inhibits the gluconeogenesis process. Second one is glucagon and glucocorticoid. They favor the pyruvate carboxylase enzyme. They activate pyruvate carboxylase enzyme. In this way they favor the gluconeogenesis process. This is the first way. Second, through covalent modification. The covalent modification it increases the production of cyclic AMP that activates cyclic AMP dependent protein kinases enzyme that ultimately activates the gluconeogenesis enzyme. In this way they favor the gluconeogenesis. The third one is allosteric regulation. Allosteric regulation, the acetyl CoA it inhibits pyruvate carboxylase enzyme and then inhibits the gluconeogenesis process, while fructose 2,6-bisphosphate it activates pyruvate carboxylase enzyme and favor the gluconeogenesis process. In this way, regulation of gluconeogenesis takes place with the help of hormones through covalent modification and through allosterically regulation. Insulin inhibits pyruvate carboxylase enzyme. In this way, inhibits gluconeogenesis process. Glucagon and glucocorticoid, they both activate pyruvate carboxylase enzyme in this favor gluconeogenesis process. Covalent modification, they increases the production of cyclic AMP that activates cyclic AMP dependent protein kinase enzyme that ultimately activate gluconeogenesis enzyme and favors the gluconeogenesis process. Third one is allosteric regulation. Acetyl-CoA, it inhibits pyruvate carboxylase enzyme, and fructose 2,6 bisphosphate, it activates pyruvate carboxylase enzyme and favors the gluconeogenesis process. So friends, it is all about gluconeogenesis, the definition, important aspects, sites, the steps and regulation. If you have any query, you may ask in comment section. And if you like the video, then kindly share and subscribe to my YouTube channel. Keep watching and thank you.